Welcome to AP Biology, topic 2.11, Origins of Cell Compartmentalization. My name is Paula Dunger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. In this lesson, I will discuss some similarities and differences in compartmentalization between prokaryotic and eukaryotic cells, and then talk about how the mitochondria and chloroplast evolved from previously free-living prokaryotic cells via endosymbiosis, and share with you some evidence to support this claim. There are two main cell types that make up living organisms, prokaryotic and eukaryotic cells. Both of these cell types have plasma membranes as shown here. So you can see the plasma membrane circled in the prokaryotic cell in figure 1 and the plasma membrane of the eukaryotic cell in figure 2. And the plasma membrane functions to separate the internal environment from the surrounding environments. Now, the prokaryotic cells lack internal membrane-bound organelles, and so the only membrane that the prokaryotic cell has is that outer plasma membrane. However, prokaryotic cells do have internal regions, such as the nucleoid region shown here, that contains its genetic material. Eukaryotic cells, on the other hand, do have more extensive internal membranes and membrane-bound organelles that compartmentalize the cell. And the genetic material is contained within a membrane-bound nucleus. And this is a distinguishing feature of eukaryotic cells from prokaryotic cells. So how did these membrane-bound organelles that we see in eukaryotic cells evolve? Well, the nucleus and other internal membranes like the ER are theorized to have formed from the infoldings of the plasma membrane. So you can see those structures on this initial cell. Mitochondria and chloroplasts, however, evolved from previously free-living prokaryotic cells via endosymbiosis. So I'm going to use the mitochondria here and its evolution to talk you through what endosymbiosis is. So a free-living aerobic prokaryote was engulfed by an anaerobic cell through endocytosis, and that's shown here in this diagram. So we have this aerobic proteobacterium that was taken in by the anaerobic eukaryotic cell through endocytosis. Then the engulfed prokaryotic cell, however, did not get digested by the engulfing cell. So it was actually more of a win-win situation. So the aerobic bacteria that was ingested took care of oxygen that was available, and the anaerobic cell could have provided food molecules, and this would lead to energy and metabolic reactions that would have benefited this cell. And then over time, the engulfed cell lost some of its independent functionality, however, and became the mitochondria for that eukaryotic cell. So you can see that labeled there. And it was a similar evolution for the chloroplast, except in this case, a free-living photosynthetic prokaryote was engulfed by another cell through endocytosis. And again, it was another mutually beneficial relationship here where the photosynthetic bacterium that was engulfed would provide energy for the cell, and the cell could provide protection for that ingested photosynthetic bacterium. But again, over many generations, the engulfed cell lost some of its independent functionality and became the chloroplast of eukaryotic cells. So what is some evidence that supports the evolution of mitochondria and chloroplast by endosymbiosis? Well, let's take a look at the relationship between the functions of endosymbiotic organelles and their ancestors. So we have the mitochondria and chloroplast here in these figures, and both mitochondria and chloroplasts have double membranes. So that's the first piece of evidence. These double membranes function to regulate the passage of materials into and out of the cell and help maintain a stable internal environment. So you can see here in these figures, they have these two double membranes showing how they were once free-living and then engulfed through endocytosis, which would lead to that double membrane structure. And like prokaryotic cells, mitochondria and chloroplasts both have their own circular DNA, which encodes genetic information, and they can reproduce by similar processes. And so you can see here that circular DNA in both of those structures. And also, both mitochondria and chloroplasts contain their own ribosomes, as shown here, which make proteins for the mitochondria and chloroplasts, similar to how prokaryotes have ribosomes to make proteins for them. So now let's practice the skill of argumentation. And in this part of argumentation, after a claim is made, it's important to support a claim with evidence from biological principles, concepts, processes, and data. So here's an AP multiple choice question for you to practice the skill of supporting a claim. So hit pause, and when you have an answer, press play again, and we'll see what you got. Okay, so the question's asking the mitochondria found in most eukaryotic cells and contain their own DNA and ribosomes that are similar to those typical of many prokaryotic cells. Which of the following statements is justified by these observations? And the correct choice is B. An ancestral cell most likely engulfed anaerobic prokaryotes in a relationship that proved beneficial to both cells. So there's that mutual symbiotic relationship that came about. Some key takeaways here are that both prokaryotic and eukaryotic cells have external plasma membranes. However, whereas the prokaryotes only have internal regions where specialized structures and functions can occur, such as the nucleoid region containing the DNA that I showed you previously, eukaryotic cells have additional internal membrane-bound organelles to compartmentalize the cell and help with cell efficiency. According to the theory of endosymbiosis, a previously free-living prokaryote was engulfed by another cell through endocytosis, and after living together symbiotically for some time, the once free-living prokaryote lost its independent functionality and gave rise to either the mitochondria or the chloroplast. And there's several pieces of evidence supporting this claim, such as the presence of double membranes in both mitochondria and chloroplast, circular DNA in both those cell parts, and ribosomes as well in both the mitochondria and the chloroplast. So thank you for watching, and I hope this video is helpful.